In this part, I'm going to talk about mechanisms of translation. Hi, now we will talk about mechanisms of translation. Translation. So what is translation? It assembles a protein using the information in the mRNA sequence. Basically, mRNA is being held by the ribosomes, and another type of RNA called transfer RNA brings in the appropriate amino acid corresponding to the message in the mRNA. So basically it brings in the appropriate amino acid corresponding to the codon in the mRNA. So it turns out that different types of RNA play a major role in translation. In general, there are about 4 major types of RNA in the cell. There is messenger RNA, and we already talked about it. This is what codes for the protein. There is ribosomal RNA known as rRNA, and transfer RNA known as tRNA. Although all of these 3 mRNAs are important in the process of translation. They all participate in the process of translation, and we will see how in a few minutes. So, but basically what happens, ribosomal RNA makes up ribosomes, the large ribosomal subunit and the small ribosomal subunit, which are scaffolds for translation. They serve as a scaffold for translation. So they hold the mRNA, and then the transfer RNA comes in and brings the amino acid which corresponds to the codons present or found in the mRNA. There's another type of RNA called RNAi. So the endogenous form of RNAi or RNA interference is known as microRNA or miRNA, and the ones that are synthesized in the lab are known as short interfering RNA or siRNA. So microRNA controls gene expression. Before we move on and talk more about translation, let's talk about microRNA. So microRNAs matter in development and human health and disease. So let's look at this example. MicroRNA we first discovered in C. elegans in the worm when they mutated a gene coding for microRNA LED7. This caused premature rupture of the worm. So microRNAs are also encoded by genes. Yes, genes can code for mRNA, but there are some genetic sequences in the genome which code specifically for microRNA. So think of microRNA as a kind of a policeman in the cell. So when you have a mutant mRNA which is produced, microRNA can bind to it and mark it for destruction so that it's never translated into protein. So here we have a kind of like a short explanation of microRNA and how they are produced in the cell. So first, a specific genetic sequence, which we think of that sequence normally as junk DNA because it doesn't code for mRNA, but it actually codes for microRNA. So we have some sequences in the genome that code for microRNA, and this results in a larger microRNA molecule. Then an enzyme called dicer chops it into a smaller fragment called microRNA, and now microRNA recognizes and binds to a specific mRNA transcript through complementary binding and marks it for destruction. So this is a way that a cell can prevent mutant mRNAs from being translated. For example, in clinical studies, they found that certain microRNAs are associated with the severity of the blood cancer B-cell lymphoma. So when a certain microRNA was present at low levels, this resulted in a more severe outcome for patients who had that, for patients with B-cell lymphoma who had this low microRNA levels. So sometimes microRNAs, or most of the time, they're called RNA interference. In the movie Hannah, they talk about a girl created using DNA interference, but they really need a better science consultant in Hollywood. So it should really be called RNA interference, not DNA interference. This is actually now available on Netflix. They changed the actors and they have it as a Netflix show. I guess Netflix has a show about everything, even RNA interference. What is short interfering RNA? So the idea is that if we have endogenous microRNA, we have endogenous policemen in the cell that bind to mutant mRNA and mark them for destruction. So the idea was, what if we create short interfering RNAs, and we can create them specifically to deactivate certain mRNA, and in this way we can potentially create drugs, RNAi based drugs to cure cancer, for example. So in about 2006, when I was in graduate school, and RNA interference was just discovered by Craig Mello from Worcester, University of Massachusetts Medical School in Worcester. And he actually got Nobel Prize for his discovery. So this was a big thing in 2006, 2005. Everyone thought this was a really cool way to turn off gene expression. Basically, for example, for my graduate school dissertation, I also worked with siRNA. We had a construct, a DNA construct that had siRNA in it, and it was delivered into the cells, and this allowed to turn off the expression of a protein that we were interested in. But actually, this technology proved to be largely unsuccessful. There are no known approved drugs, I believe, that are based on siRNA, but there are some drugs in development. So it's still relevant to biotechnology. So this is again the mechanism of RNAi. It interferes with gene expression, so you get no mRNA, and as a result you get no protein. It opens new possibility to design drugs made of siRNA or based on siRNA. So again, the mechanism is such that a gene encodes RNA, which ends up looking like this double-stranded RNA. A dicer enzyme comes in and cuts it into short or microRNAs. There is something called an RNA-induced silencing complex called the RISC in the cell. It's an enzyme that attaches to siRNA, and siRNA RISC complex ultimately attaches to target mRNA. So here's our target mutant mRNA, and chops the mRNA into small pieces. So now let's come back to the process of translation. There are 3 types of RNA involved in translation: mRNA, ribosomal RNA, and transfer RNA. Messenger RNA encodes the amino acid sequence. Ribosomal RNA serves as a scaffold during translation, and transfer RNA transports specific amino acids to the ribosomes for protein synthesis. So let's talk about mRNA. It carries information that specifies a particular protein. 3 mRNA bases in a row form a codon, which is something that we talked about earlier. A codon specifies a particular amino acid. Most mRNAs are about 500 to 4,500 bases long, depending, and this will determine the size of the protein. So larger mRNA means that the protein will be longer normally. mRNA can also be called a transcript. Information in the transcripts are used to produce a protein. So what is rRNA? rRNA or ribosomal RNA form ribosomes. Ribosomes consist of 2 subunits that join together during protein synthesis. They provide structural support, and some of them have catalytic activity or enzymatic activity. There is a hypothesis that all life from Earth stems from an RNA molecule because RNA have enzymatic activity. So here we have a picture of the small ribosomal subunit and the large ribosomal subunit, and they're called that because of their weight, which is determined by the cesium gradient, and you can see that one subunit is larger than the other one. The small ribosomal subunit holds the mRNA, and the large ribosomal subunit holds the incoming transfer RNA that brings in the appropriate amino acid. So what is tRNA or transfer RNA? Think of it as a translator from the language of mRNA or from the language of nucleic acids to the language of amino acids. So tRNA is the middleman. It binds to the specific mRNA codon using its anticodon domain and brings in the appropriate amino acid. So it has a shape of kind of like clover leaf shape, and it has 2 ends. One end contains the anticodon, which is complementary to the mRNA codon, and the other end bonds to specific amino acid. So here we have the picture of transfer RNA, and specifically it has the anticodon. So for example, if mRNA says AUG, tRNA has an anticodon that says UAC, so it's a complementary match to AUG, and it brings in the appropriate amino acid. In this case, it's a methionine. Now let's look at the next codon as GGA. It's CCU as an anticodon and tRNA, and it brings in glycine. So there are 61 different species of transfer RNA or tRNA, and each one is specific to a particular amino acid and has a specific codon. Just like in the table, we have 61 codons and 3 stop codons. So this is why we have 61 different species of transfer RNA. So you can see here's the anticodon on this loop 2. This is the mRNA binding site, and this is the amino acid accepting end. So this, again, just to review the central dogma of molecular biology. DNA makes mRNA makes protein. A codon in mRNA codes for an amino acid. This is our table with 61 codons coding for specific amino acids and 3 codons which are stop codons and do not code for any amino acid. The genetic code is universal. Different codons that specify the same amino acids are termed synonymous. So if we go back to the table, proline, CCU, CCC, CCA, CCG, they all code for the same amino acid called proline. They are called synonymous codons. They are only different in position number 3 in the codon. Non-synonymous codons encode different amino acids. So the code is a triplet code, and DNA has a certain reading frame. So when there's a disruption in the reading frame by adding a base or deleting a base, this can really create a mutation. So these mutations are known as insertions or deletions, and they can disrupt the reading frame, and this is known as a frame shift mutation in the protein. So here's an example. If you have an mRNA sequence, GAC, 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 and so on, one base is added. This will disrupt the reading frame and alter an amino acid. 2 bases are added. Once again, this will disrupt the reading frame and change the amino acid sequence. But when 3 bases are added, this preserves the reading frame. Yes, you have an extra amino acid present, but the rest of the amino acids are correct. So this mutation would be less severe than the other 2 because it preserves the reading frame. An example from a test, from the upcoming test, if you have 3 patients who came to the hospital, they all have mutations in the same gene. Gene has a G insertion. Dave has UG being inserted in his gene at this position here, and Martha has UUG inserted. Who has the least severe mutation of the 3? It's Martha, okay, because she does not have a frame shift. She just has an insertion of 3 nucleotides. So translation, how does it actually occur? It requires mRNA, tRNA with amino acid ribosomes, and energy and protein factors. So translation is divided into 3 steps: initiation, elongation, and termination. During initiation, you have the mRNA and the tRNA, which has met amino acid attached to it, binds to AUG with its anticodon. And so in the second step, small ribosomal subunit comes in and also binds to the mRNA. This is known as the initiation complex. Now let's talk about building a polypeptide. Now the large ribosomal subunit attaches. It has 2 sites, a P site and an A site. The P site bears growing amino acid chain, and the A site holds the next incoming amino acid to be added to the chain. In this example, we can see that the first amino acid was methionine, and now we have a codon called GGA. So the next tRNA comes in, bringing in amino acid glycine, and it has an anticodon CCU, which corresponds to GGA. through complementary binding. So anticodon complementary binds to GGA. Amino acids, once the amino acid glycine comes in, methionine and glycine get joined together by a peptide bond. So the incoming amino acids get joined together by a peptide bond. And once that happens, the previous tRNA leaves, and so in this case we can see glycine is leaving and cysteine takes its place, and then the new tRNA carrying a lysine comes in, and they keep producing this chain of amino acids. So the amino acid chain gets extended. Each piece is brought in by tRNA, whose codon corresponds to consecutive mRNA codon as the ribosome moves down the mRNA. How does translation get terminated? This occurs when a stop codon is encountered by the A site of the ribosome. The protein release factor frees the polypeptide. The polypeptide is the same as saying a growing chain of amino acids. So a chain of amino acids is called a polypeptide because amino acids are joined by peptide bond. So it's a polymer made of amino acids joined by peptide bond. That's what a polypeptide is. The ribosomal subunits then separate and are recycled, and the new polypeptide is released.
Translation is initiated by formation of an initiation complex consisting of the smaller ribosomal subunit, the first amino acid tRNA, and messenger RNA. The larger ribosomal subunit then joins the complex. Proteins called initiation factors are also involved but are not shown. The 70S ribosome has two sites to which transfer RNA carrying amino acids can bind. One is called the peptidyl or P site, and the other is called the acceptor or A site. The initiating transfer RNA carrying its amino acid binds to the P site. A transfer RNA that recognizes the next codon and carries the second amino acid then moves into the A site. The amino acid carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site. The ribosome now advances a distance of one codon and the first transfer RNA is released. A transfer RNA carrying the next amino acid now moves into the A site and the ribosome shifts down by a distance of one codon. This is followed by the two amino acids on the transfer RNA in the P site being transferred to the third amino acid. The ribosome continues to move along the messenger RNA, and new amino acids are added to the growing polypeptide chain. Elongation of the polypeptide is terminated when the ribosome reaches a codon that does not code for an amino acid, called a stop codon. The ribosome dissociates into the smaller and larger subunits, and the messenger RNA and protein are released.